So, Mac, welcome to Talk Python to me. Awesome to have you here.
Hey, thank you, Michael, for the invitation. I'm happy to be here. Excited.
Yeah, I'm pretty excited to be talking about medicine and uh, all the stuff that you guys are doing with Python. And I really like these kinds of shows because I think it's important to highlight that Python is not just for web developers and pure data science machine learning people, but it's used by this huge spectrum of people doing all sorts of interesting stuff and solving real problems with it, right? And you sounds like you fall pretty solidly in that category.
Yeah, absolutely. It was like Python has been sort of this discovery as I've gone through my career as a physician, and it's interesting how to begin with, even computer science initially when I was training and kind of growing up, it was hard to imagine medicine and computer science sort of being hand in hand together. But now I think things have progressed, and there's a lot of technology that's now in medicine that allows you to do all these fascinating things. And of course, as I've discovered Python, it brings in kind of the toolkit and the ability to to be able to achieve and solve problems in a way that I think it's not been envisioned before. So it's a very exciting time.
Yeah, it is a very exciting time, and I think it's just getting better and better. Before we get too far into this, tell people a quick bit about yourself, quick introduction.
Yeah, absolutely. So I'm Somak Roy. I'm a molecular pathologist. It's a type of physician who deals with looking at the genome of either a patient or a patient's tissue, and we essentially look at all of these things in a way to be able to help manage patients' treatment. In my current position, I am an associate professor and the director of molecular pathology at Cincinnati Children's Hospital. My lab is a clinical lab that is under the division of pathology, and we do a lot of work that pertains to kids in terms of helping them diagnose and manage pediatric cancer, as well as infectious disease that happen in this age group. um, Molecular pathology. I essentially trained back in India as a physician, did my MD, then came here, started in Pittsburgh. I did my training in pathology and lab medicine. I specialized in molecular pathology. Then I was there in Pittsburgh. I worked for some time and then opened Cincinnati Children's.
Excellent. Do you work directly with patients, or do you get samples sent to you from other doctors and then you process them and analyze them?
Yeah, that's a good question. So I do not work with patients directly. It's a subspecialty in medicine where my lab works with the samples that have been collected in the patient, either from the OR or a procedure or from the radiology suite, and then we work on that tissue or the blood sample or a bone marrow sample that comes to us. And yes, then all the testing that we perform is off from that specimen. And then once we generate the clinical reports back, they go back to the patient's chart and to the patients to the clinicians who are treating and managing them. And then that way it helps how they're able to then get a diagnosis and then give the appropriate treatment and the management to the patient.
Yeah, excellent. So yeah, you must see a lot of different stuff flying through the lab you have to analyze. So how did you go from I'm studying medicine to I'm writing Python code and running automation and what was that process like?
Well, that was an interesting journey for me. So before medicine and biology came into my life, I started off. It was second grade, I believe, when my dad, he got me a computer at that time, which is a 64 kilobyte small machine. So it was, I think, this was Toshiba MSX computers, where you could write like GW basic code and some basic predefined hex code, and you can run small applications on that. That was my starting point. It was super exciting for me, and I think from there on, the journey went into, as I went through school, high school and then college. that medicine was, I would say, biology was something that intrigued me. And at the same time, I also got interested in genetics, looking at DNA sequences, and just I had a natural sort of liking for the fact that I could study about the cell, about the genome, the DNA and the RNA, but also I realized that there was a lot of math and computation that you can use to sort of slice and dice the data. And at that time, the place where I grew in India was a very small place, so we didn't have access to resources like internet. So my exposure to internet was when I actually went to med school for the first time. Like, oh yeah, you can actually connect to like other computers. So that's when I started my medicine. So obviously I did my training in medicine in medical school back in India. That's when I started to connect and talk to a lot of people, and some of my friends who actually were already writing apps at that time using like Java applets on browser. And so I started to make some connection in terms of images, and how learning how some of those things can be used in medicine. And so radiology, during my radiology rotation, that was my first real life it was my realization that actually in medicine you can use computers a lot to handle a lot of these images, X-rays, CT scans, and I think as it went on, when I came to the US, that's where it really started off. During my residency in pathology here, I actually connected with my mentors, Dr. Anil Parwani and Dr. Leon Pentanoids. They are well-known pathology informaticists. They've spent a lot of their time sort of dwelling in the world of pathology, medicine, and computer science. And so that is when I could actually realize that yes, you can do a lot of innovative stuff by developing apps, algorithms, analyzing either image data or molecular data. And so that is when I started to get into designing an app, it's a very simple web app that was a project I was working with one of my mentors. And so the idea was that we had a lot of these pathology images, and we wanted to create a little in-browser app that could display these images as thumbnails, and then clicking that could enlarge the image, show it in display. And so I used at that time was .NET framework to be ASP.NET, and so create a little app using Visual Basic. Slowly, I then migrated to using C# in the same environment. And that time, I started my advanced fellowship training in molecular pathology. That's when when I started there. That's when I realized there's a lot of genomic sequencing data where essentially you're dealing with a lot of strings and numbers, and you have to make a lot of sense in terms of this large volume of data that comes in.
If we're working with so the kind of data that you're working with for, say, this genetic stuff.
Yes.
For when you're studying the genomics, so how much data is in, say, one strand of DNA? How much of that do you actually care about? Like, give us a sense of sort of how much data we're talking.
Right. So it really depends on what is being done. And so when we look at, when we talk about genomics, it is really designed on how the experiment is done. So for example, if we just simply look at the entire human genome, we are talking about 3 billion alphabets. Essentially, it's the combination of 4 alphabets, A, T, G, and C. So these are the 4 nucleotides of the DNA sequence, and the RNA has one additional one which replaces A. But the idea is that it's a mix and match of these sequences. And so if you think about the entire DNA, the entire human genome as a single thread of A, T, G, and C's in various combinations, you're looking at 3 billion alphabets. And so what happens is when we do these sequencing experiments where you would take the DNA molecule from a bunch of cells within a tissue, and then either we read all the 3 billion base pairs, and typically the way the sequencing is done is you read all of these sequences and from many molecules, and so you'll have multiple copies of that when you're translating from a molecular, like a chemical molecular structure, to a DNA sequence on, say, a flat file on a, in a file system. And so if you look at that large scale of data, like the entire genome, we are talking of, hundreds of gigabytes, maybe even terabyte worth of data. Then there are other
Wow.
more practical approaches when we look at the genome. And especially this is something that we use for day-to-day patient care, which is referred to as targeted sequencing. What that means is instead of the 3 billion base pairs, we focus on those regions of the human genome that are of most pertinent use, or that we, at least as the current field of genomics, that we understand what to do with it. And so there are certain genes that, at least in the in the space I work with, cancer genomics, there are, I would say, close to about maybe to 1,000 to 2,000 genes which are known to be cancer associated, and of that, roughly about 500 to 700 genes are where we know that they've been studied and demonstrated that if there are certain types of abnormalities in those genes in terms of the sequence changes, that they have certain meaning from in context of a tumor, in order to make a diagnosis, or to understand if the tumor is aggressive or benign, or if there are certain treatments that could be applied to those tumors. And that's specifically linked to the kind of sequence change you see in that region of the genome. And that, we are talking about, practically speaking, when we talk about the targeted testing that we do, it's a very small fraction of the large genome. Typically, there's a term known as exome sequencing, and exome sequencing refers to sequencing all those regions of the human genome where it at least encodes for one or the other annotated gene. That is typically about 1 to 2% of the entire genome. And so if we further narrow that down to about, say, 500 to 600 genes that one would typically sequence for practical cancer molecular testing, I would say that's probably about a tenth, or maybe slightly less than that, of the genome, but it's a very high yield from a clinical standpoint, because
Sure.
the most alteration you will find that would help with the clinical treatment is high. So if you talk about that data set, that is, it's complex in a different way, because just looking at the raw sequence data would be, somewhere in the, I would say 1 to 20 gigs from a single, sequence file. But it entirely depends on how deep we go. So, for example, when we talk about sequencing, as I mentioned before, when we sequence a molecule, we can sequence it either at certain depths. That means what level of redundancy you want to be able to read that molecule. Sometimes we read the molecules 20 to 30 times, so that's referred to as 30x. Or sometimes we'll read that 500 times, so that will be 500x.
Do you do that because you want to make sure you don't misread the?
Yes. So
gene?
right. So what happens is, the greater the depth of sequence. So typically for such large panels that we sequence in a clinical setting, we usually target about 1500x to 2000x. That means we're reading that 2000 times. So the more the depth it is, the possibility of identifying a certain variation or genomic alteration that is present at a very low level. For example, say you have a tumor cell, and within that, only 2% of the cells have this mutation, others don't. And so when you're looking for or hunting for these needles in a haystack, you really want to maximize the amount of depth you have to be able to pick those things up. So it really depends on how deep we go. The more deep we go, the more data it is, and so it can scale up to almost several hundred gigabytes.
Sure. Yeah, I've always wondered about how you can go and read somebody's genetics and then not make a mistake when you're reading, using chemicals to read just a whole bunch. It's really ridiculous how much data is there.
Right.
Off by one, a C for a G or whatever is a bad thing, right?
Right, but it is, I think as the technology has matured, there's nothing 100%, in terms of the error profile for the enzyme that has been used to work, the technology that is reading the actual fluorescence, converting that to signal. There's always statistical values and probabilities that are associated with what is the probability that it is wrong or incorrect or correct. But within that frame and where the current technology is, it's pretty accurate for if not all, many of the regions of the genome. And so
Excellent.
it's mind baffling how it works.
Yeah, it really is quite amazing. It's one of the modern marvels of science for sure.
It is. It is.
Yeah. So I think you're a little bit unusual, a little bit weird in the sense that you got into as your first sort of programming thing to bring to apply to your science and medicine side of things was C# rather than or VB.NET rather than something like Python or
Yeah.
R or something. So maybe talk a bit about that experience, contrast it with Python. Like why do you end up moving to Python?
Yeah, so I think the reason I started using VBNet C# was, I would say, most it was probably influenced a lot by, at the time when I was doing my med school in India, as to what was available at that time. It was not something I would just go to the internet and like start getting a lot of resource as one would do now. So it was pretty much like, this is the book I have available, and that's about .NET. So, okay, that's the only thing. So you start.
Yeah, so this is what we're gonna use. Okay.
Right, and then, but the thing is, when I started applying C# and it was mostly C# and a little bit of C++ that I started to get into, like with some of the non-genetic stuff initially. Well, the project I was working on, it was not too bad because I was able to accomplish most of the task. But then once I got into genomics and I came, so the way professionals who get into genomics and molecular pathology, there are a couple of different routes. So either they're physician, people who are physician trained and they kind of have a formal background in medicine, and they kind of do a specialized training, and then they become molecular pathologists after getting board certified. There is the other route, which is more of a research background, where sort of people have spent a lot of their time in really deep research. They've learned a lot of genomics hands-on, either from a computational background or from a more laboratory, like a wet laboratory background. And so they've spent, they've obviously done their PhDs and postdoc training, and then sort of come into the molecular pathology field. People starting there tend to have more of a formal computational training. So they're getting, they usually get, obviously, when you start with a research lab, R, Python are sort of like the most common tools that are used for any kind of data analysis and data set visualization. Coming from more of a foreign medicine background, typically when we get training in clinical informatics or pathology informatics, often it is very kind of I would not say corporate, but it's a very formal application development space. So it's a lot of Windows based, sort of, .NET, C#, C++, that kind of thing. So
standard
Java,
enterprise stack. Yeah, Java or .NET is a probably likely choice. Yeah. Okay.
Yeah, in bioinformatics, I mean, at least in genomics bioinformatics, kind of the ecosystem of tools available, it's a mishmash of everything. There's for anything which is very computationally intensive, like when you're trying to align sequences to the human genome, those are very intensive tasks, and typically it's a lot of C++, Java that's involved in some of these very mainstream tools that are available. More recently, I think we are seeing Rust coming into the picture as well. There's some Golang applications, and then of course, Python and R are like the predominant I think tool sets or the programming language that are used to solve a lot of these problems. So when I sort of started my molecular pathology fellowship and I got into, now I had to do this project that involved manipulating all the sequence data to a point where we would be able to develop an application that could help sort of, it's a web-based application that could help, for other pathologists and faculty to read that sequencing data and, digest in a very way that's easy for them to look at it, rather than going to the Linux, terminal and opening up like, raw files and things like that. So I used, that was my first project, was to use C# in that context. But I quickly realized that there was a lot of these algorithms that were natively either written in R or Python, and then having to, incorporate those functionalities was not as easily possible. So I had to rewrite a lot of those things in C# primarily. it was a good learning curve, but I think from a maintainability perspective, it was getting really difficult. And so
Sure.
that's when the realization was that I think a combination of Linux and Python was, I had to move towards that.
Yeah. C Sharp, probably from the time frame that you're thinking about, didn't really have a great package manager story, not to the same degree that Python does. Although they do
Yeah.
pretty good now over in the
Right.
.NET land.
Right.
Yeah. All right. So, good question from Chris in the audience says, is there a reason to use Python specifically? Like, are there some special sauce packages that make it attractive? It sounds like that's kind of what you were getting at. Like, you found more solutions to these algorithms than available in Python than in C Sharp or
Yeah.
whatever languages. Yeah.
Right. So I think, I think the simple answer is yes. I think the community and the amount of work that has been done in this particular space with genomics. I mean, when you are really searching for applications, it kind of falls into these three categories of, anything which is a high performance compiled, program that is usually in the Rust, C++, C, a lot of, of those languages, a little bit of Rust and Java. And then the other bin is essentially kind of split up into Python and R. I think for me, Python was, and I think I'm sure others have shared the same way, where it's almost like, wow, this is amazing. Like coming from C Sharp, it was a little bit of a change because there's no more curly braces and think about the whole indentation and things. But I think
Did you miss your semicolons?
Kind of like even
I
now,
know.
sometimes when I write like a little bit of JavaScript, I feel like, oh yeah, okay, this semicolons here, but or like
Exactly.
the curly braces. But not too bad. I think what I got onto was like the simplicity of the language and how powerful it was when, like, if I'm thinking about, it was interesting when I had to do something like there was an algorithm where I had to parse out certain, strings in a way where it required some known workflows that we use to do like variant annotations when we are cross-referencing databases and putting them together. When you look for in terms of like C# packages, there's really nothing there for it natively to it. So you have to write a lot of those things. In Python, it's the amount of time that you spend in developing those things is much faster. Like the development time itself is quick because you either get an idea of somebody who's already done the work or there's a more formal package that you can use. So I think initially when I started off, BioPython was a very interesting collection of package. It was like a tool suite essentially written to to have all these functions available for very common day-to-day tasks. I want to query certain region of the BAM file, or I want to parse out certain things in the FASTQ file to look at some of the sequences, or doing counting number of sequences in a given file and getting read counts, things like that. It's all out of the box. And so that was sort of like the first thing, like, oh wow, this is amazing. I can just say somebody's already done the work and just
instead of
putting on top of it. So
yeah,
it was very
instead of creating these, I'll just like use those. Perfect.
right. So that was one. And the other motivation to use Python was, say for example, why not R? Why Python? Because R also is a very rich ecosystem in at least in genomics and visualization. So I think the second thing was in terms of the idea that I was working on was having to develop a web application and all of these bioinformatics, toolings and algorithms running sort of in the backend. And so at that time, it was like, okay, well, Python I've not heard much about in terms of web application. Mostly it was, again, this big, like, C# .NET. That was why I started off, with that. But then at that time, there was Django, and then Flask was sort of coming in. It was a very minimalistic, sort of application. So I started focusing on that. It was very easy with Flask to, get up and running with very simple, applications to do that. I didn't try much into Django just because it was too bloated for me. But Flask was great. And then what I realized was you can create a simple web application, but then at the same time you can use all your BioPythons and all the wonderful bioinformatics packages in the backend. So it's like a single language that lets you do both. And so like, this is great. It's just I don't have to go anywhere to learn a third or a fourth or a fifth different programming language, and this just gets the job done.
Yeah, keeping in mind that your main job is medicine, not programming, right? It's not like you're a
Yes.
CS person who's just out to learn all the languages, right?
Right. So that definitely is, and again, that's a huge, I would say, as you said, I'm sort of in an unusual position where I'm, a physician, but I also do a lot of these application development. So that certainly is an important point in terms of how much time I have to be able to develop these prototypes. And then obviously, typically the way it works is, at least right now here in, where I am currently working, I have an excellent and amazing team of developers and bioinformaticians who really do a lot of the development work on the front end, back end. And so for me to be able to take the additional time out of my, the clinical and the patient care work is limited. So if I can get whatever prototype I'm thinking of or developing the application fast, then that's what I'm going for. And so
right, then you can hand it off to the team and let them polish it up and make it production ready, basically.
Yeah.
I was wondering how much time of your job do you get to spend on these kinds of things, finding new packages, optimizing or improving the ways that you're working on stuff versus just sort of handing it off to the folks you work with and keeping, focus more on the medicine side.
Yeah. So that I think it's a good question. I think it's evolved over time as I've being sort of when I was in training and then being a faculty and then faculty in this new position. One of the things I did was, as part of my certification, was to to get board certified in clinical informatics. That's a discipline by itself that involves a lot of it's a very broad field in terms of informatics and healthcare, and then one of the buckets there is software development. And so I was I was quite interested sort of in that field. And so most of my time in terms of being able to devote to finding new packages or trying to write up an application that could solve a problem or coming up with prototypes, it was done in a way that sort of aligned with the work I was doing. And so it would be days when I'm on clinical service where I'm mostly working on sort of with patient care related matters. So those weeks would be obviously very busy. I would have I would wake up at like extremely early in the morning, spend the first 2 hours, 4 to 6 a.m. just working on this, and then I get back to like the clinical work. And then there would be weeks when I'm off clinical service, so I'm not responsible for any patient care related work, and those weeks would be where I would spend time in terms of doing these investigating into sort of some of these packages and coming up with new ideas, exploring what is all what is available in terms of certain problems that I was solving. And that time sort of my quote protected time professionally was spent in that. And so that would be maybe a week spent into like, hey, we are trying to look into this variant annotation tool, and then we want to write wrappers around it so it becomes easy for our lab's operation to be able to use that. And so kind of that how it works. So some of those either early mornings or the weeks I'm off clinical services, how that works out.
Yeah, excellent. Is it changing fast? So I'll give you an analogy, then you could tell me about your space. So on one hand, in Python web world, you mentioned Flask and Django. Flask and Django, while they are evolving, they're kind of the way they have been, and they're pretty stable. And if you learned Flask 5 years ago, you're still good to use Flask today.
Mm-hmm. Yeah.
Or is it more like Fast API, Pydantic, message spec? Just there's something new all the time that you got to keep learning to bring in. Are there a ton of new packages just coming online, or is there a set of really solid ones?
So I think it's both yes and no, and so it depends on what what area we're working on. So right now in the clinical lab that I'm directing here, when I came here in 2020, it was when we started off from scratch. So essentially, the idea was to be able to bring up a pediatric cancer sequencing infrastructure that was not available, and so it was ground up from the lab to personnel to space to computational infrastructure, everything. And so we kind of have this sort of two big bubbles in that operation from an informatics perspective. One of them is we essentially are in the process of developing our custom lab information system. That's essentially a web app. And so we have that space, and the other space is bioinformatics. And so bioinformatics is a lot of the custom scripting or the applications we develop is Python based. Some of them we do with Golang, when we need a little bit of performance aspect. And then the other aspect is the web app. So from a web app perspective, when I started here, we actually started we use FastAPI and Python. So that's kind of that was so the idea was that, well, since we're starting from scratch, and I came to know about FastAPI at that point of time, the whole thing was about acing away. I was pretty much sold on that aspect. And then I think the whole tool set made a lot of sense. I'm like, okay, well, this is perfect time to be able to. I think when I started, FastAPI was 0.5 or 0.6, and so now obviously you can see a lot of change happening there. So yeah, that definitely is a lot of fast pace, and so we kind of do catching up in a sense where it has to be done in a careful way. The reason is because from as compared to more traditional research lab testing where at the end really there's a lot of discovery, there's a lot of excitement. At the end it all translates into being sort of the data is presented at a conference or you publish that as a manuscript, and that's the end point. So if you move off from version 1 to version 3 of an algorithm, you have to obviously make sure that your research, everything is reproducible, but beyond that is not a problem. But when we're talking about the same thing in context of a clinical care for a patient, the room for error is very, very little. You can't make mistakes. And so the entire space of clinical testing is very regulated in that sense, because there's a lot of requirement that you have to perform that any change that has happened in your pipeline, say you're using some version of an application, now you upgrade to a newer version, you have to demonstrate that the analytical performance in terms of sensitivity and specificity for that pipeline didn't change. And so a lot of work is needed when you go do like a version upgrade. So we keep those things very controlled and and careful, versus some other things which are more in the R&D space. There's a little bit more room to play around with tools.
Right. Yeah. Chris was asking in the audience a great question about basically, is it more exploratory and you just move really fast and don't really worry about tests and stuff like that? It sounds like this is more of a production type thing. Like if you're going to run it over and over, and if it gives a different answer at some point for testing for a disease or something that's really bad, you need it to be right all the
Yes,
time. And so,
yes. So the
yeah.
rule when we do new test developments or we bring a new algorithm, obviously that part of which we refer to, there's a formal term that we use in lab medicine. It's called familiarization and optimization, or ONF phase. That's where there's a lot of flexibility, new tools, new version, trying out different things. But once it moves from that into the validation phase, and then once we deploy the application, once the deployment is there, it's a production application. We don't touch it unless something really has to be tinkered with, or there's a bug that we have to fix.
Who's in charge of running those apps? Is that people on your team in your lab, or is that the hospital, or how?
Yeah, so the way it is set up here is, so when I started off, I was the N of one. So I started off with the FastAPI application. I had to build up we had a bioinformatics pipeline that I had initially authored. But then when we went through the validation phase, I luckily had two people on staff who kind of were handling the bioinformatics on the front end, and then eventually we had a third person who joined the team. So then they were kind of helping me out with a lot of the actual groundwork of writing the code, getting tests done, going through the validation data, summarizing that for me. Being a lab director, it is my responsibility ultimately to sign off on all those things. Say, hey, okay, if this is the validation and this is what is being demonstrated, that your package or your pipeline or whatever you're working on demonstrates this level of sensitivity, then yes, I, being a lab director, say that yes, this is working. And once that happens, so we then deploy those applications into production. We use GitHub and the usual dev, test, prod cycle, and so that's kind of how it works.
Cool. Do you have your own hardware, or do you put stuff like on DigitalOcean or AWS, or?
So
You say?
with healthcare data, there is generally a little bit of angst with data sitting on the cloud outside the premise of our institution.
Yeah.
I would say like the institution where I work on is very that way it's quite uh, very forward thinking in being able to use modern technology. So what we started off, and since it was everything being built up from scratch, we are taking the decision to keep things on prem for beginning, but we also kept in mind that at some point of time, if the institution decides that, oh, we're going to switch our infrastructure to using AWS or Azure or whatever the platform is going to be, that we want it to be ready. And so the way we had it set up, and this is due to our amazing IS team here in at our institution. So we had our own hardware that we got in terms of the actual servers, and I had we collaborated with the IS team to be able to help us build our Kubernetes infrastructure. So we have a test and a prod Kubernetes cluster, and then all our apps and the bioinformatics pipeline. Well, the apps for now and the bioinformatics pipeline that we're looking forward, in the near future to get deployed on these things. As a matter of fact, what we've done
Okay.
is our dev team, we do a lot of the development on Kubernetes as well, and then we keep moving all these things as containerized applications.
Oh, that's excellent. So really embracing containers and Docker and Kubernetes, and that should make it super easy to move to wherever you want to go, right? Anything that
Yeah.
can run Kubernetes, you just push to that and you're good to go.
Right. I mean, that's it's it's a little bit difficult to start with, but I think once
Yes.
we are in that stream, then it is much less effort to move things around.
Yeah, last year I rewrote all of our servers and APIs and condensed 6 to 8 servers all into one just Docker cluster, and it was a great decision. But to me, it was also a little intimidating to like, well, here's one more thing I have to manage and understand, and if something goes wrong there, then everything else still breaks. And but
Right.
having it set up is really nice once you get used to it.
It is right.
All right, let's talk a bit about. Well, I have a question for you. I want to talk about some of these packages
Yeah.
that you've been saying are like a lot of the reasons you chose Python and you use a lot, which is great. But before we get there, like I got the biopython.org website pulled up, and the very first line is BioPython is a set of freely available tools, open source, freely available.
Right.
How much does that matter to you guys? On one hand, you have a ton of money being in the medical space. It's really high stakes, so paying for commercial software or commercial libraries is probably not the biggest worry. On the other hand, open source is really nice. Being able to look inside is really nice.
Right.
Free means you don't have to deal with getting permission. How does that fit into your world? I know how it fits into like small startups and things like that, but
Right.
for a hospital, for example, what does free and open source mean to you guys?
I think it does have a lot of impact in terms of how we end up working and setting up these things. And obviously, whatever I'm speaking is representing what it means from sort of an operational standpoint. When we talk about molecular pathology, generally being able to bring up a clinical service like that is a huge investment. And so a lot of the investment is, and this is generally applicable to any institution where something like this is been set up for patient care or clinical use. The investment is primarily in a lot of the instrumentation and the reagents that we use are generally quite expensive, which is sort of I would say when we talk about what is the cost of a test when it is offered. That cost factors in a lot of these, operational costs that we need to, buy these expensive sequencing instruments, the reagents that are used as consumables as we do the test, over and over again every week, every month. So from that standpoint, traditionally, the way things have been designed is, I would say 10 years back, when we would, work with our finance team to say like, okay, the cost of the test is going to be so and so based on all of these different inputs. And so, 10 years back, computation, bioinformatics, all of these were not factored in at all. But now, as we are in that era where, using GPUs on a regular basis to be able to do, simple, I would not say simple, but, routine work to get from the raw sequence data to be able to identify genomic variants, that's getting common. Using FPGAs, using large clusters to be able to, to perform these tasks. And so now we are starting to see those costs getting in as part of the ultimate, cost that goes to the patient for a test. And so we try to minimize those things. One of the way to be able to minimize those things is to be able to choose between free open source versus something which is a commercial product. And it's always a balance between the reliability and the service that you're able to get back, saying, hey, something breaks down, we know, there's an SLA, there's a certain, assurance that this thing is going to, will have help, versus open source free would be where we feel very confident in the code base. We sort of, sometimes what happens is when we, when we kind of use some of these open source tools, we end up almost invariably having some wrapper around it to change things, or being able to have some insight into the source code. So it depends on that balance, what we choose.
How often do you fork it and use your self-maintained version versus just run what is publicly on PyPI and then maybe wrap it to orchestrate it a bit?
So I would say for the web application part of it, we don't really do a lot of forking. We kind of go with what it is. The only thing what we do is, since we have the luxury of using a combination of GitHub and containers, and knowing the fact that the regulatory requirements require that you tightly version control all these things with, history and all those things, we tend to, when we are developing these things, when we are validating it, before we do that, we try to stick to a fairly stable version. So for example, things like, beta or release candidates, we try to stay away from that, even if they have some desirable features. But unless we see like a full production, version of that, we don't tend to switch to it. So we keep things like that without maintaining or without forking or kind of, making up modifications. When we get into more of the bioinformatics stuff, where we are actually trying to use an algorithm to solve a particular piece of part of the pipeline that is doing some data transformation, it depends on how much we want to change or modify. That's when we, sometimes fork it. Sometimes we fork where we know that, and this is kind of the unfortunate reality in many of these scenarios, where, you have great open source tool, but after some time, due to whatever, financial or business or other reasons, that they stop maintaining it. And so essentially we kind of get into this freeze mode. We tend to fork that so that at least we have that available. And then if we make any changes, that we kind of, keep it to that fork. But generally I would say it's probably in the 80-20, where 20% is where we fork it, make some change. Most of the times we try not to do that. But yes, open source free tools have a big impact. A lot of the tools that we use as part of our bioinformatics pipeline, as a matter of fact, which is kind of used in the community of molecular pathology to build these bioinformatics pipeline, they tend to use a lot of open source tools. And the reason for that is, for example, it's not written in Python, but we have there's an algorithm called BWA. It's written by Professor Heng Li. That's one of the algorithms that is almost like a de facto, I would say, when it comes to doing sequence alignment. That's a part of the pipeline. And so it's a tried and tested application for more than a decade now. So really, there's not and this is a fairly stable, algorithm or application. So we don't tend to, it's well maintained, from an open source perspective. So those obviously are we highly rely on those. But, there is this whole ecosystem of softwares that come under this rubric term of variant calling, where we're trying to identify these different variants. There's a whole bunch of those, and some are fairly well maintained. They are open source. Sometimes, depending on the context you're using, you need a license if it is used in a commercial setting. You don't need a license if it is in an academic setting. Like for example, when we do clinical testing in a institution such as where I am right now, that's an academic institution. So typically it's not, it's not for profit. And so obviously we, we don't need licenses for that use. But once it goes into a pure commercial space where if the lab is, doing all of this testing for profit, then, there's a license requirement. So we see a combination of these, things showing up it's actually becoming more common now with open source tools that at least in the genomic bioinformatics space.
Yeah. Oh, excellent. I think another benefit probably for you guys, I know it's a benefit for a lot of organizations, is if you use the open source tools and you need to hire somebody new, there's a good chance that they have experience already with those tools. Whereas if you use something private, expensive, you might have to teach them from scratch what the thing is, right?
Yes, that is correct. As a matter of fact, it's fortunate that a lot of the people who've done a lot of good work and have contributed to the genomics bioinformatics space have, the general tendency is whenever we are setting up any sort of, pipelines for, DNA sequencing, RNA sequencing, or, more from a research perspective, methylation sequencing, single cell RNA-seq, UMI-based error-corrected variant calling. There's a very thriving open source space, and so that really helps with people who come in, even if they're not familiar with these tools. It's easy to get familiar with because there's a lot of community backing that up. Or as you said, when we hire people who already are coming from a different lab, or they've had some experience, but they come and say, oh yeah, we know how to do alignment, or I'm aware of these applications that use that. It is much easier from a learning curve perspective rather than having to now open up a manual and this is the proprietary thing that only works here. And yes,
exactly. Cool. All right. Well, we coordinated a bit on a list of packages that you've used in your lab or find really helpful for your work, and maybe we could touch on those just a little bit.
Yeah.
So CNV kit, genome-wide copy number from high throughput sequencing. I don't know what that means, but tell us about it.
Yeah, absolutely. So CNV or copy number variation, this is a type of genomic alteration where what happens is, in a simplistic way, at least when we talk about cancer, the cancer cell, sometimes for it to be able to survive, it tries to use different ways of doing that biologically. One of the way to do that is certain genes that help a cell to grow in the absence of nutrients or with very little nutrition. Is certain genes, if it has more copies of those genes than normal, then it will, okay, it's like you have more money than expected, you can do a lot of things. So typically what happens is, in a normal human genome, any cell that we pick up, it will only have 2 copies of the gene. One is coming from your mom, one is coming from your dad. In cancer, what happens is, in certain scenarios, if a gene that helps with growth of the cell, or it helps the cell to survive even without signal or nutrition, if it has more copies of that, it will make 6, 8, 20, 50 copies, it can survive. Versus there are certain scenarios where if there is a gene that is supposed to regulate the cell so it doesn't go haywire, if the cancer is able to delete one of the genes out, then you only have 1 gene left. You knock out the thing, and then that protective mechanism is gone. So then the cancer cell can easily survive. So what happens is, with CNV or copy number variations, the idea is that we use the high throughput sequencing data to be able to infer how many copies of these genes do we have. Is it more than 2? Is it less than 2? And so this particular package, it's a very, very well-established, well-maintained package in the community that essentially does this thing. Is you give it the sequencing data and define the regions of the genome that you're interested in. You can also provide like names for the regions, like this region is gene BRAF, or this is EGFR, or whatever you're interested in. And then what it'll do is it'll do all the analysis to be able to tell you that, okay, well, when we are comparing this particular tumor against this reference set of 20 normal samples, where we know that you should only have 2 copies of the gene, in this particular tumor we are seeing there are 50 copies of the gene. So it gives you kind of an output data that numerical can tell you that what it does is it does a log2 based transformation of the data, saying that okay, after all of this computation, when I compare to the normal, this is 50 times more, or this is 20 times more the expected copy, or it is half of the amount of copy we need in terms of deletions. So that's what really it does. And it's written in Python. It uses a lot of Python. It has Python dependencies that that have been written in sort of in either C or Python C bindings. But at the end, it gives you that data, and it has an internal visualization tool. But I was not very happy with how it was written, so I ended up writing a wrapper which is called CNA Plotter. It's open source. It essentially uses the end data for from CNVKit, and then it gives you a nice visualization of the copy number. So I think if you go down, if you scroll down, there's some example images.
Yeah, you have this on GitHub, so people can, if people want to use this, it's right there, right?
Yep, it's right there. Yeah. So I think at the very bottom of the images, where the screenshots there.
Oh yeah.
Yep, right here. Yeah. So for example, the first image over here, you can see this. It's a thin band of all these multicolored things, and each one of them is a single human chromosome. So chromosome 1, 2, 3, 4, so on and so forth. And if you look in the image, it is at the Y scale essentially is log2, which says 0, and going up it is 1, 2, 3, and then it's a negative scale on the lower side. So anything going above 0 means you have more copies than 2. Going below is less than 2 copies. And so if you see here in this example, the plot here, you see the very end, which says chromosome X. Is a single, the band over here is lower at -1. That means this is a male patient with a single X chromosome, as compared to females who have two X chromosomes. And so when we look at this plot below here, this is actually a plot from a cell line, a tumor cell line that is abnormal. And here we see there are two genes which are amplified. One of them is a gene known as TERT, and the other gene is MDM2. So these two genes are again one of those examples where it gives the tumor survival advantage over other. And so you can see here there are multiple copies of these genes as compared to the baseline over here.
I see. So that might predict something like how survivable the cancer is, or
yeah, like how aggressive it is, right. So if is it going to be localized, say where it happened, or it's going to spread to other parts of the body, or be difficult to treat, or be resistant to treatment? Yes, things like that.
So if this is you want higher numbers, not lower numbers.
Well, it all depends, right? I mean, certain genes are good genes. For example, if there are certain checkpoint genes, if those numbers, if they have lower numbers, you want to have 2 copies of them, because if that protective mechanism is gone, the tumor becomes very aggressive, again, depending on the tumor. So it is all into context. So if you're looking at the good genes, you want to have 2 copies of the good gene. If you're looking at some of the bad genes, you don't want to have more than 2 copies of the bad.
So 1 or 0 is better. I got it. Okay.
Yeah.
Okay, HGVS.
Yes, this is again a wonderful package that was initially, I think it was started by a person named Reese Hart. I think he still maintains it, but there's a lot of it's a very well publicly maintained open source package. There's a lot of community involvement in that as well. So what HGVS is, it's a nomenclature system for giving a name to all these variations. So when you talk about, I'm not sure, Michael, if you've heard about the term mutation. So mutation is a very commonly used term that refers to some kind of abnormality in the genome. In this case, so what happens is there are these standards that most clinical labs follow when they're putting all of this information in the patient report, saying, okay, this particular tumor has a mutation in BRAF, a mutation in EGFR, some other gene, and there is a certain way that those mutations are described in terms of what sequence alteration is happening, say at the mRNA level, and what sequence alterations are happening at the protein level. So now in your protein, you're missing these amino acids, or you have excess of these amino acids, or something got switched from here to there. So there is a formal way of defining that, and the guidelines of the group that defines that is referred to as HGVS. Human Genome Variation Society. And so it's a very complicated process where you have to do all these translations from the genomic scale, where the numbering system starts from 1 to like, and whatever the length of your chromosome is in terms of ATGCs, and each chromosome has a different number. And if you have a certain alteration that is happening, say in chromosome 7 at this particular position, then you have to translate that to the mRNA of that gene and then the protein of that gene. So it's a lot of math, a lot of strings involved in that process. And so essentially this HGVS Python package provides all of those functionality as a wrapper. You can create your translation. You can essentially project the variant from your genomic to the mRNA to a protein level, or vice versa. You can validate things. So we ended up. I actually wrote a paper about this when we did a validation of how well this particular package works. And so now, in the lab that I'm currently in, we implement this thing for generating those nomenclatures. So what happens is when we put a report out in the patient's chart, and when our say our oncologist who's treating the patient, they want to know, okay, what did you identify in this tumor genome? They would read that nomenclature saying, oh, okay, this particular change in this BRAF gene, this is significant. I know that there are therapies that are out there that we can use to treat this patient tumor. So that's what this nomenclature system is about. So it's a very
Nice.
complex but fairly automated system.
Yeah, and it normalizes it if there's multiple ways to represent it and things like that. Very nice.
Yes.
All right. This one I'm familiar with, OpenPyXL.
Yeah.
I guess you probably have a lot of data that either comes from or gets shared out into Excel, right?
Yes. So what we do is we sort of are right now in our lab, we're kind of in this sort of kind of an interim phase where we sometimes use Excel to look at some data. So traditionally speaking, before typically any lab that goes from 0 to the point where you have a web application that automates everything, the intermediate phase is using a lot of Excel files. So it's very common in many labs to use Excel for a lot of different things. For QC, for charts, for tracking. So we use this OpenPyXL for a few things. One of them is when we have a lot of the sequencing data that we have to summarize and then generate a QC to be able to present that, to essentially create an Excel document on the fly from the backend to provide that whatever data they want to look at in terms of statistics or list of variants or some form of calculation they want to do further. That's where we use this package. Typically we use it as part of our bioinformatics pipeline when we have to generate those things, but it's a very handy tool. We actually use something similar, and I'm forgetting the name of the package, that is used to generate our document. Like we use some Word documents for creating reports, but we also use Python there to be able to summarize a lot of these data points and then create a Word document that it starts with a template of a Word document, and then use Python to fill up all these
right. Here's where the graph goes. Here's where the summary goes. Here's where the detected whatever goes. Yeah.
Right.
Yeah. Cool. Here's two things that overlap. Are you familiar with this thing where scientists rename human genes to stop Excel from misreading them?
Oh yes, absolutely.
Oh my gosh, this is crazy.
Yes, this happens. When we import a lot of this data coming from somewhere, we'll see entries like September 14th or March 19th. These are not
Yeah, this is a big problem going in and out of Excel. And so as much as you can do in Python or any proper programming language rather than using Excel. But there was one that was MARCH1 or March 1.
Yes.
Or SEPT1.
Yeah, exactly. It's very funny. Some of the gene names are funny, but then Excel gets it to the next level when it changes the names.
Who put a date there? This doesn't make any sense. Yeah.
Yeah, it doesn't make any sense. Yeah.
All right, on to the next one. Hera.
Yes, Hera. This is very interesting. So this is where I think where in our instance we are going away from standard web applications, standard bioinformatics pipeline to really touching DevOps using Python. And so one of the things that typically we get to the point when we scale up our bioinformatics pipeline where we have multiple samples and multiple runs and everything needs to be orchestrated in a way where you have, while you're running your pipeline, you have a lot of visibility into how it works. And so this is one of our projects we're working on to move our current bioinformatics pipeline, the way it works, kind of on a single server to be able to use the Kubernetes cluster to actually deploy the long-running pipelines onto that. And so there are many options. There are a more standard sort of WDL-based protocols that you can use to run on cloud or HPC environments. There's a very popular tool called Nextflow that is used to be able to kind of create your data analysis PyFlow. You can sort of define that and then use any backend to deploy it. One of the things that we kind of were, when I was exploring the space, one of the things I came across was the whole sort of ecosystem that Argo maintains with Argo workflow and Argo CI/CD and all those things. So workflow was interesting because Argo provides that way where you can sort of write your pipelines in a YAML format and then have it deployed on the Kubernetes cluster. It really is very native to the Kubernetes cluster.
Interesting.
So,
it sounds a little bit like Ansible, but specifically for bio type of projects, right?
Yeah. So Argo. So interesting thing is Argo, when Argo workflows were set up really for a lot of CI/CD automations in mind. So it is, yes, you can run data pipelines in general, but it was never, at least in its description, it never describes the use case sort of in bioinformatics or biology pipeline analysis. And similarly, it was like, okay, it's a generic tool, you can use it for whatever you want. So I tried it out with using like a YAML file, and it was a simple 4-step pipeline. It was wonderful. It was magical. And the good thing was with Argo, like with the Argo workflow, when you install that on your Kubernetes cluster, it comes with a native web interface. So it's I'm sure if you've heard about the workflow option with Airflow. So Airflow is a package that also there's a nice Python SDK for that, where you have you deploy it on a Kubernetes cluster. We have all these amazing visualizations to show what step you're on, or if there's some error there, it'll do that. Argo does the same thing. So it has obviously the built-in capability to interact with it as an API, but then also there's a web interface that it'll deploy, and you can have visibility into every step of the process. You can summarize, you can see the entire tree. So that was very interesting for us because we could get all that thing done in a single go. But then our challenge was, our desire was that if we could integrate that with our LIMS system that we're working on using Fast API. So the idea was, hey, if there's any Python SDK, and so that's where Hera comes in. So Hera essentially is a SDK or a wrapper that's essentially talking to Argo, but you can define your pipeline steps as DAGs in Python. And so that makes the process so super simple, where you're now natively, essentially, we can integrate that as a backend to our web application. And so then it's almost like, it's Python again from start to finish. You're not, getting out of that. And it's again, it's a very well-maintained application. So we are currently doing a validation to be able to make sure that, or demonstrate that, it's equally performant when we compare to a more sort of native shell-based, execution of the pipeline.
Okay, yeah, this is new to me. I mean, of course, I know Airflow, but not Hera. Cool. All right, PyInSim. Did I grab the right one here?
No, I think it's similar. Let me see if I can send you the link.
Yeah, throw it in the private chat here, and I'll pull it up.
Yep. Here. Okay.
There we go. InSim, not PyInSim. Yeah. Okay, got it.
Yeah. So this is a very interesting space in next generation sequencing assay, or for high throughput sequencing assays. So what happens is As I mentioned, that one of the things that's required for a clinical lab is to be able to perform a validation on multiple samples of tumors that have certain mutations, and then you can demonstrate that, yes, the assay works because you have tested, 100 samples that have 300 different, mutations or genetic alterations, and then you can demonstrate that, yes, your pipeline or your assay was able to pick it up. So you can say that, your assay is, X percentage sensitive, X percentage specific, and, your recall rate and things like that. So what happens is when we're trying to get those samples that have these very difficult or challenging variants to detect, because they're just, complex in how they occur biologically in the cell, is very difficult. Some of these are very rare. There may be only 2 samples in the entire world, or it's just not possible practically to, get those samples unless we wait for like, 10 years to, validate that. So the idea is that if it is possible to be able to use algorithms which can manipulate the existing sequence. So for example, we have a sequence data from an existing real tumor sample, but we can manipulate that in a way where we introduce these mutations in silico. So we can introduce, SNVs or insertion deletion mutations, and then use that same file to then feed into our pipeline, bioinformatics pipeline, and say, okay, run through the entire data pipeline, and at the end, let's see if we are able to identify those variants that we inserted. And so that's where
Oh, I
this whole
see.
in silico mutagenesis comes in.
Cool.
It's a very hot topic. It's a very relevant topic of interest to really fill this large gap in terms of availability of rare variants and rare samples, and how we can really improve sort of some of these rare but very clinically significant edge cases where we don't want to miss those variants and actually see those in real tumor samples. And so this is a Python package that was developed by at the University of Chicago as part of their clinical lab. And so what really it does is it will take in a list of different, mutations. For example, in this plot, I think they give examples of, insertion, deletion, insertions where you have extra sequence, or deletion where you have certain sequence which are missing, or SNVs or single nucleotide variants where you have one nucleotide that got switched with another one. And so these are typically that we practically see in like real samples or real tumor samples. But this is a way to mimic that, in a sample that does not have it. And so this Python package is able to, take that list from, you say, okay, I have a list of these 20 important mutations that I know from the public databases have been reported, but I want them to be inserted into my data set that was created from, say, a set of 3 or 4 real tumors, and then use that to challenge the pipeline to say that, hey, can you still pick it up? And so
I see.
that's what this does.
Simulate these rare changes and then sort of test or exercise your setup. Cool.
Yeah. Right.
All right, we got a few more to cover, but I think we're getting a little bit short on time. So let me just close this out with a final question for you, because I know this is the topic du jour. What does AI and LLMs look like for you guys? Does it matter? Is it really powerful? Is it super important? I mean, genetics is kind of text data in a sense, and so it's sort of in the space of how it could apply.
Right. It is a text data, and it's a lot of there's when you talk about like a search space, a lot of the search space is very text based. There is some numerical based, but there's a lot of text based search as well. And I think across the entire spectrum, from where we start with very raw sequencing data to the point that we are trying to ask the question that, okay, I found this rare or novel mutation in this particular gene. What does it mean? What tumor has been described? What disease does it relate to? One of the things that we do as molecular pathologists, and this is sort of where a lot of the medical work comes in, is where we really go through a lot of the medical literature, what we have learned before, new publications, papers out there that that have a lot of data in terms of studies that have done on this particular gene, and they've described like, okay, these alterations actually activate the gene, or is bad for the tumor, or makes it treatment resistant. So you can see that naturally there's a lot of text search that happens. And so in that space, we are seeing I would say in the past 3 to 4 years, there's been a lot of application of AI tools that have come out. Particularly in the space of variant calling, where we have this genomic sequence data and we're trying to identify variants. One of the examples that's been talked about a lot is the deep variant caller. It's called deep variant from I think the team at Google who developed that. That uses a lot of the AI techniques to be able to pick those things up. There are some genomic databases that we use for in silico prediction. For example, if you have a variant, we have no idea about it. It uses there's a database called DBSCSNV that uses random forest techniques, and I think it uses another algorithm to predict if a certain site where there is a mutation can enhance abnormal mechanism called splicing versus not. Similarly, there's a lot of tools that are coming in, and the LLMs I think are, I would say, not mainstream, but I think there's a lot of interesting research that is coming around there where people are trying to use LLMs for doing these more broader text search, saying that, hey, I have these, I don't know, 1,000 articles, and I want to find these particular combination of words that, there's a combination of a disease and a mutation, and what do I get back out of that? I personally tried, ChatGPT with different, phrases and questions about it. What I've seen so far is, and this is purely my personal experience, I think a lot of it reads very real, but when you start to look into the references as to what it references, then you quickly figure out that
just making stuff up, right?
this is not the real deal. And so I think it's I'm not a very pessimistic person. I would say, oh no, this is all garbage, but I think there is opportunity there. It's just how do you train it?
Sure, maybe there's a space or an opportunity, and it probably already has been. People are pursuing this is
right.
Training a smaller model, but really deeply in genetics, whereas not trying to use a model that tries to understand everything.
Right.
Yeah.
Is more in the medical literature or the genomic literature to be able to like the training is enhanced in that. Yes. So I think there's active work going on there, but it's I think it's making a lot of interesting research, a lot of potential impact on how we do things, and obviously the tool sets that we currently use, we might expect in the next 10 years to change.
Yeah, for sure. All right. Final thought here. People are listening. They're maybe doing similar work to you. How do they get started? What would you tell them? Get going with Python and some of these packages.
Yeah, I mean, I would, my reflecting on my own experience, sort of, in a very winded way that I ended up here. I think, Python is, I feel programming in general, and I think Python particularly as a programming language is a very low, entry point in terms of being able to really quickly get things done, like learn it easily and get things done. I think it should be, to me, anybody who's trying to pursue something in biology or computational biology or bioinformatics, I think this is the first thing. It's something easy to do, to be, I would say relatively easy to do, to be able to get into that. Anybody with a desire to learn this, has analytical thinking. I think investing into Python is probably the best bet, because you can pretty much do anything you want. That's what I tell, when I train people in my lab, or I talk to other students, is that if you want to spend your time, you have very little time because you're busy, with your other things. I think the one thing that can get some of the job done and be still aligned with what you're doing is Python. And after that, I think it's a lot of self-driven learning where you kind of looking into things. But the good thing is, I think the Python community is wonderful. It's almost like I sit down and I think about, I have to solve this problem. Probably there are 50 other people who are thinking about that, and maybe 2 people have already worked on it. So it's
right. They've already published it to PyPI, and you're good to go.
Yep.
Absolutely. I totally agree with that. And people should take the couple of weeks, get good at it, and it'll save you time, definitely, in the long run.
Oh, absolutely, yes. The only thing the only thing I would say, like an added thing, is if somebody is learning Python and then they do have an intention, to take it to the point where they would be involved in more serious, application development, or maintaining an open source package, or however they contribute to that. I think learning a little bit more, like learning Python in its real sense in terms of how to do it right. There are 5 ways of doing something correctly, but I think there's one way that is consistent so that it's, again, easily shared, it's easily maintainable, others can easily understand. I think that would be my second advice. It takes a little bit of time, but I think it's well worth the effort to spend the time on writing, idiomic Python code. So it's portable.
Absolutely. All right. So, Mac, thank you for being on the show. It's been great to get this look inside to what you all are doing with Python.
Yeah, thank you for having me on the show. I appreciate that.
Yep. Bye.